Good morning, everyone. It's my great honor to present today a transformative topic at the intersection of immunotherapy and RNA technology, enabling in vivo CAR-T applications through circRNA. I'm Jingjing from YXgene, and we will dive into how this innovative approach is poised to overcome significant limitations in current cell therapy paradigms. This preparation will guide you through the fundamental principles of circRNA, our advanced manufacturing and purification processes, and the compelling preclinical data and support its therapeutic potential. Our goal is to showcase how circRNA can pave the way for safer, more effective, and truly off-the-shelf CAR-T therapies, moving us beyond the constraints of traditional ex vivo manufacturing. Let's begin with the biogenesis of naturally occurred circRNA. CircRNA are unique class RNA molecules distinguished by their covalently closed-loop structure. They are generated intracellular through a specific process called back splicing, where a downstream 5' site joins an upstream 3' site. This biogenesis pathway results in a molecule that lacks of 3', 5', and 3' characteristics of linear mRNA. This absence of terminating is not merely a structural curiosity. It's the key feature that confers upon circRNA their remarkable stability, as they are inherently resistant to exonuclease that rapidly degrades linear RNAs. This innate durability forms the basis of their therapeutic advantage. Furthermore, the mechanism of translation initiation differs fundamentally from linear mRNA. While linear mRNA relies on a file cap structure for ribosome recruitment, circRNA translation is driven by an internal ribosome entry site, or ARIS, enabling cap-independent protein synthesis. This mechanism bypasses the dependency of cap chemistry and instability issues brought by poly-A components during manufacture process, offering flexibility on multipurpose expression control. And the therapeutic benefits of circRNA are multifaceted. Firstly, the exonuclease resistance grants it a profoundly longer half-life compared to linear RNA, enabling weekly and persistent protein expression rather than mere days. And secondly, this stability guarantees not only longer but also higher and more consistent level of protein production, which is crucial for effective CAR-T cell function. Thirdly, the circular structure and careful sequence design significantly reduces recognition by the innate immune system, leading to lower immunogenicity and minimizing inflammatory responses. This robust structure also contributes to superior thermal stability, simplifying storage and logistics for clinical application. This slide perfectly encapsulates the value proposition of circRNA for CAR-T therapy. It illustrates a clear evolution. First, mRNA CAR-T already offer advantages over traditional viral DNA-based methods by being a true off-the-shelf product with no risk of genomic insertion. Now, circular mRNA is overcoming the inherent limitation of linear mRNA. Specifically, circRNA provides improved pharmacodynamic and pharmacodynamic properties, enabling a much longer pharmacodynamic half-life for sustained CAR expression. And coupled with is lower immunogenicity. circRNA LNP complex can be administered in vivo to potentially generate functional CAR T cell directly within the patient's body, revolutionizing the treatment paradigm. Translationing from concept to reality requires a robust and scalable manufacturing process. The preparation of therapeutic grade circRNA begins with in vitro transcription or LAT, where a laminar RNA precursor is synthesized. The critical next step is circularization, where this laminar precursor is enzymatically ligated into a closed circle using our proprietary ribozyme-based technology. This reaction mixture then contains a pool of molecules, the desired circRNA, but also leftover laminar precursor, nicked RNA, and other impurities. This necessitates a highly effective purification strategy, which is the cornerstone of producing a highly quality product suitable for therapeutic use. Apart from isopath strategy, which utilizes azo-ACASE group 1 intrin ribozyme, however, at YXgene, we have developed and explored our proprietary platform, FlexCirc. This technology is based on terahymnic group 1 intrin ribozyme. A key differentiator of FlexCirc is that its structure developed not sequence dependence. This provides unparalleled flexibility and efficiency in designing circRNA constructor for various therapeutic payloads without being constrained by specific sequence requirements. This innovative approach is protected with a patent already being submitted. Designing the circRNA construct is as important as circularizing it. To ensure high and stable expression of encoded protein, such as a CAR, we employ sophisticated optimization strategies. These include meticulous codon optimization to enhance translational efficiency and stability with human T cells. Furthermore, we utilize our proprietary RNA-seq platform for the design. This involves careful selection and engineering of the various element that drives independent translation, as well as optimizing the overall secondary structure of the circRNA to ensure it is not only stable but also high translatable, maximizing the yield of therapeutic protein. And purification is a critical differentiator in circRNA production. The standard method often involves RNase treatment, which degrades laminar RNA but leaves circRNA intact. However, this method has limitations. It shows a preference for shorter sequences and less compact structures, potentially leaving behind impurities or efficiently purifying longer and more structured circRNAs. In construct, chromatographic purification developed by YXgene is a superior physical separation method. It can achieve circRNA purity exceeding 90%, has no sequence length restriction, and is fully compatible with very compact RNA structures, ensuring a pristine final product. Our first generation analytical method utilizes the exclusion chromatography, or SEC-HPLC. As you can see from a chromatogram, This method is structured to achieve baseline separation between the desired circRNA peak, and the peak contains linear mixed and pre-RNA impurities. This highlighted the need for more advanced analytical and proprietary method to ensure the highest quality standards for therapeutic development. Well, a step forward, it was clear that we needed higher resolution for both analysis and purification. This slide demonstrates a significant leap in analytical capability. The National Institute for Food and Drug Control developed a reverse phase HPLC method that beautifully resolved the three key components: circular RNA, nicked linear RNA, and linear pre-RNA into three distinct, well-separated peaks. I'm pleased to report that YXgene's next-generation analytical methods achieve a similar high fidelity separation profile, remarkable improvement over our first-generation chromatography. This advanced method is under continuous optimization to provide the most rigorous quality control for our RNA products. And this implementation of our advanced chromatography has transformed our manufacturing output. Our process is highly efficient, following IVT and circularization, which itself achieve a 50 to 80% efficiency rate for construct between 1 kip and 5 kip. We employ a one-step chromatography purification. This process consistently yields circRNA with a purity greater than 90%. As rigorously qualified by RP-HPLC. Furthermore, we achieved the impressive volumetric yields of 8 to 12 grams per liter of reaction mixture, with an outstanding recovery rate of over 60%. This efficient and scalable process is engine of our CDMO services. The true test of any platform is in the data. In a directly head-to-head comparison, the advantages of circRNA become immediately apparent. We delivered both circRNA and the standard linear mRNA, both encoding luciferase and packaged in LNP. The true test of any platform is in the data. In a direct head-to-head comparison, the advantages of circRNA become immediately apparent. We delivered both circRNA and standard linear mRNA, both encoding luciferase and packaged in LNP. Intramuscularly to bulk mass. The imaging data taken just 3 days post-injection shows a dramatic difference. The circRNA treated subject exhibit a strong and sustained luminescence signal, including robust and lasting protein expression. To contrast, the signal from the linear mRNA is already significantly damaged, and highlighting its transient nature and potential for immune-triggered clearance. This virally underscored the superior pharmacokinetic and lower immunogenicity of our circRNA platform. And finally, to enable in vivo CAR-T therapy, we need a precision delivery system. Our solution is antibody-targeted lipid nanoparticles, or APLNPs. These are LNP decorated with antibodies designed to home in specific cell surface markers. Our in vitro data is highly promising. Using this targeted LNP, we have achieved highly efficient transfection of primary human T cells isolated from PBMCs. As you can see, we have effectively delivered payloads to both CD4 and the CD8 T cell subsets. This demonstrates the potential of using a targeted RNP system to deliver circRNA encoding a CAR construct directly to T cells within the body, bypassing the needs for complex ex vivo manufacturing. So what does YXgene offers our CDMO partners? We provide an end-to-end solution built on foundation of robust IP and technical excellence. First, we offer well-developed circularization strategy, including our proprietary flag circ technology, with assured freedom to operate for major regulatory markets. Second, we provide optimized codon and array selection strategies to ensure high-yield protein expression. Third, we have established superior purification strategies, delivering up to 80% pure circRNA. And fourth, we employ a stable and reliable purity determination method for QC. And finally, all this is supported by our GMP-compliant quality management system to get your product to the clinic. And thank you for your time and attention today. In summary, circRNA represents a powerful and versatile platform that directly addresses the key challenges of durability, immunogenicity, and manufacturability in the field of RNA therapeutics and the in vivo cell engineering. We believe this technology hold the key to unlocking the next generation of accessible and effective cell therapies. We are eager to explore potential cooperation. Please don't hesitate to contact us via email and connect with us on LinkedIn. Thank you.